Sure, my pleasure. Hi, everyone. My name is Amelia, and I had the absolute pleasure to work with Sarah Ovadio this year. Sarah is an amazing, responsible, and committed student who's very passionate about medicine. She is very interested in anesthesiology and pharmacology. As you can see, these are very huge specialties, but together we were able to combine both and came up with a fun project that I think that you will all enjoy. I now hand it over to her. Sarah, you're up.
So I'm Sarah Badia, and I'm in 11th grade. And I decided to research the blood-brain barrier and how it gets affected when someone has a seizure. Next slide, please. Next slide, please. So I had the honor of working with Dr. Amelia Halleck. Dr. Halleck is a physician and a researcher, and she's leading all clinical research activities in the gene therapy program for the bone marrow transplant division at Cohen Children's Medical Center. Next slide, please. So whenever deciding what to research and what project we should choose, I mentioned that I was interested in pharmacology and also in learning more about epilepsy. So we decided to focus more on the blood-brain barrier because when someone has a seizure, it affects the blood-brain barrier, and certain medication could also pass through the barrier, which could also help reduce or manage seizures. So we decided that the blood-brain barrier was going to combine with pharmacology and epilepsy to create the perfect topic to research. So the blood-brain barrier is a membrane that protects the brain from any foreign substances. So it only allows certain things to enter and certain things to come out of the brain. So it prevents any harmful medication or toxins from entering while allowing certain medication that's going to be helpful to the brain to pass through the barrier and reach the brain. Next slide, please. So for this research project, I learned about the structure and permeability of the blood-brain barrier and how the structure is affected and differs in someone with epilepsy. And we were able to recreate an environment that's similar to that of the BBB for our demo. Next slide, please. So the blood-brain barrier is composed of capillary endothelial cells. So the endothelial cells are in the capillary, which is like a small vessel, and those are connected by tight junctions. And these tight junctions only allow certain substances to enter and exit through the cells. So as I said before, it only allows certain helpful medication to enter, and it prevents any foreign substances from reaching the brain. Next slide. So as I stated before, I was interested in pharmacology. So a main question about the blood-brain barrier is why some medication is able to pass through and reach the brain and why some isn't. Next slide, please. So the tight junctions are what allows certain molecules to enter, and only allows certain small molecules to enter, and the molecules have to have two requirements. The first one is they need to have a molecular weight of less than 400 daltons, which is an atomic mass unit that's usually used in chemistry and physics. And the molecule has to have less than 8 hydrogen bonds. And if it has these two requirements, then it's considered small enough to pass through the tight junctions and reach the brain. Next slide, please. So now we're going to look at a regular cell at a molecular level. So up here and down here, we have the tight junction proteins, and those aid in the function and structure of the tight junctions, which, as I said before, is what allows certain molecules to enter. Here we have cadherin, which is what opens and closes the endothelial cells. Next slide, please. So now we're going to discuss the role of the leaky blood-brain barrier. So the leaky blood-brain barrier is when the blood-brain barrier has certain small holes and abnormal tight junctions, which could be an effect of epilepsy. So if the tight junctions are abnormal, they don't function properly, and they'll allow certain things to enter, which normally wouldn't be allowed to enter, and result in certain stuff that are needed to exit, which could result in more barrier leakage and more seizures, which is sort of like a positive feedback mechanism. So someone could have a seizure, which could result in barrier leakage, and then that barrier leakage could result in more seizures. And the abnormal tight junctions usually result in a loss of water and potassium, and it increases the drug penetration to the brain, which could often be harmful. So when speaking about epilepsy, some anti-seizure drugs are used to reach the brain and help manage the seizure. So these drugs have to be small and lipophilic and uncharged, which makes them able to pass through the tight junctions. And an example of this medication is clonazepam. So if we look over here, we see if a seizure affects the temporal and frontal lobe of the brain, which results in a leaky blood-brain barrier, that person that's having the seizure could be affected by not being able to speak properly or fainting or not being able to communicate or speak. Next slide, please. So as I stated before, one example of an anti-seizure drug is clonazepam, and that could be used to treat and manage seizures. It could be either given orally or through an IV. And as I said it before, it has to be lipid soluble, uncharged, and small enough, which is going to make it able to quickly and efficiently pass through the blood-brain barrier and reach the brain to help manage the seizure. Next slide, please. So for our demo, we're gonna have two different models. The first model is an irregular blood-brain barrier. We have the brain that's placed in a plastic bag that's not permeable, so it doesn't allow certain molecules to enter. And when we put salt in, we see that the servo on the side, which represents skeletal muscle, continues functioning normally, so that the seizure wouldn't really get affected. Like a person wouldn't have a seizure because the barrier isn't affected. But now in a leaky blood-brain barrier, we put the brain in a dialysis tube, which is permeable and allows substances to be exchanged. And we see that when we place it in water, the servo starts twitching, which represents muscle spasms. So what we used for the demo was a beaker, which we placed the brain in. We used dialysis tubing as the permeable membrane. And then we used wires and Arduinos to help connect the servo and to help measure the concentration and so that it functions according to whether the brain is in a permeable or non-permeable bag. Next slide, please. So some of the things I enjoyed about this experience was being able to be guided through this whole entire experience by someone who's in this field and was able to teach me so much. Some things I learned is that in medicine, when you go into a certain research project or anything with one mindset, you could actually find out more than you actually think you would. So you could go in just trying to research about the blood-brain barrier and come out with actually learning more about epilepsy and how that could be treated. And throughout this whole experience, I also learned how to properly conduct research. Some of the challenges that we faced were deciding how to actually create a model and a demo that displays the environment of the blood-brain barrier and how that environment gets affected when someone has a seizure. So I really want to thank Dr. Halleck for guiding me through this whole experience. Any questions?
I don't have a question. I just have a comment that you've really done such a fantastic job of demonstrating it. The demo was so nicely done. Thank you for showing something that's non-computer science so well in a biology, concept so well. Thank you to you and your mentor.
Thank you. I think go more in depth about, the question was what's the next steps about researching this topic. I think I would go more in depth about the structure of the barrier and exactly what other medication could pass through and what wouldn't, and what other diseases actually affect the structure of the barrier.